So we're going to look at some mitochondria structure and do a quick respiration review. Since we're talking about mitochondria, we might as well throw in some oxidative phosphorylation words. So here we have an electron microscope image of a mitochondria and then a diagram of a mitochondria. The diagrams are way easier to make sense of. The electron micrographs get kind of confusing. But we can go ahead and match up some pieces and parts. We've got an outer membrane. There is also an outer membrane on that electron microscope picture of the mitochondria. There's an inner membrane. The inner membrane has some folds. Those folds are called cristae. Crista for one, cristae if it's plural. The purpose of the folding of that inner membrane is to provide more surface area because this is where all of the proteins and enzymes of the electron transport chain are embedded. More surface area means more space for more electron transport chain, which means more ATP production. So again, we have outer membrane, we have outer membrane, we have inner membrane, we have inner membrane. So that, whoopsie, inner membrane, PowerPoint really doesn't like when I draw small. Inner membrane is here. The space between the membranes is known as the intermembrane space. This space here, this space here, this is the intermembrane space. Let me get an easier color. Intermembrane space because it is the space between the membranes. It's important that that space be small because this is where we pump those protons to get that hydrogen ion concentration gradient. If there was big space, it would take a lot of protons to get a gradient to be very high. If the space is small, we fill it fast. So that's the benefit of having a very small intermembrane space. We can fill it up with hydrogens really fast, which means that it doesn't take much for us to build that concentration gradient. Outer membrane, inner membrane, of course, this is the cytoplasm. And we've got here the matrix of um, the matrix of the mitochondria. This is where we're going to have some DNA. Not a fan of how it was added to the diagram here. It really would have been better to show it as circular pieces of naked DNA, because of course we know that mitochondria have naked chromosomes. You can't really see it in the electron micrography because they're very small. They just don't pop up very well unless you're staining for exactly that thing. We've also got some ribosomes. That might be what they meant for this to be in the diagram. That's not what these are. So these are not actually ribosomes. These are little crystals of enzymes and ions that build up inside the mitochondria. But there are ribosomes in there, those little ribosomes of 70S ribosomes, 80S out here, 70S in there. So again, we've got outer membrane, we've got inner membrane, we have folds in the inner membrane to make more surface area for more electron transport chain. We also have a very small intermembrane space that we can build up that hydrogen ion concentration very quickly. And we've got the matrix. The matrix is where the link reaction occurs. It's also where Krebs cycle occurs. Glycolysis is out here in the cytoplasm, and then electron transport chain oxidative phosphorylation happens across that inner membrane. So a little bit in the matrix and a little bit in the intermembrane space, kind of going back and forth across that inner membrane. That is some structure. Let's talk, actually, let me go back one. So we're going to zoom in on like this one little piece. So imagine that I chopped out this piece of mitochondria and I'm going to zoom in close. This is what that little piece would look like. So again, this is going to be cytoplasm up here. Here is our outer membrane. Here is our inner membrane. This is the intermembrane space. And this, of course, is going to be the matrix. If you need to see the full picture, that outer membrane is going to go around the whole entire mitochondria. That inner membrane is also going to go around, but it's going to go around with some folds. Those folds are the cristae, so there's more surface area, so there's more of all of these things that we see embedded in that inner membrane. All this stuff is the electron transport chain. So let's talk about some cellular respiration review stuff. Of course, in the cytoplasm, we are going to have glycolysis. In glycolysis, we're going to turn glucose into pyruvate. One glucose gets chopped up into two pyruvates. We need to use 2 ATP to make that happen. Basically, what goes on is this: the phosphate groups from the ATP get stuck to the glucose. That makes the glucose be too high energy. It wiggles and it jiggles and it's not stable and it cracks in half. Just like if we were to take a cookie and wiggle it and jiggle it and crack it in half, we'd have some crumbs. When we chop glucose in half with these phosphate groups, we also end up with some crumbs. Those crumbs are electrons and protons, also known as hydrogen ions. Those electrons and protons get picked up by NAD. NAD is therefore reduced, because it gained electrons and protons, to NADH. So we're going to end up with some NADH. We're going to end up with these two pieces of pyruvate. What's kind of cool is that we also end up making in the end 4 ATP. So we use up 2, but we make 4, so we get a little bit of ATP produced in just glycolysis in the cytoplasm. The pyruvate then is going to enter, the mitochondria. It's going to end up in the matrix. That NADH that we formed is also going to get transported into the matrix. Pyruvate in the matrix is first going to go through the link reaction. In the link reaction, that pyruvate is first decarboxylated. Pyruvate has 3 carbons. We're going to chop one off. Decarboxylate. We chop off a carbon. When that happens, we get some crumbs, and again, we can make some NADH. What's left over here, those 2 carbons, we call this an acetyl group. That acetyl group in the link reaction is going to get chunked onto a coenzyme called CoA, forming, you guessed it, acetyl-CoA. That acetyl-CoA then ends up in Krebs cycle. We are still in the matrix. I know I'm like oozing out into the intermembrane space. All of this is still happening in the matrix. Krebs cycle, these carbons that we had left over, we continue to chop them off of that coenzyme A. Every time we chop off a carbon, we have some crumbs. Those crumbs get picked up by NAD. NAD is reduced to NADH every time it grabs some of those crumbs of electrons and protons when these pieces of glucose are decarboxylated. The NADH. And then we're also in Krebs cycle going to reduce FAD to FADH2. FADH2 and NADH end up at the electron transport chain. So again, I know I oozed all of this into the intermembrane space. It is not there. All of this happens in the matrix. The NADH that we make in the matrix, the FADH2 that we make in the matrix in Krebs cycle is sent over, it diffuses to the electron transport chain. Whoa, I don't know what just happened. That NADH and the FADH2, they give away. So now we are oxidizing. They're losing their electrons and their hydrogens. There's that hydrogen popped off. There's the electron popped off. The electrons are passed to the electron transport chain. The electrons jump, 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 jump. As the electrons jump, 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 they lose energy. That energy is used to take those hydrogens, the hydrogen that also popped off of NADH and FADH2. Those hydrogens are pumped. We need energy to do it. We're getting the energy from the electrons jumping. We're going to pump those hydrogens into the intermembrane space. Electrons jump. We get hydrogens off of FADH2. We get electrons off of FADH2. Electrons are jumping, jumping, jumping. The energy from the electrons jumping is used to pump the hydrogens into the intermembrane space. So now we've got all of these hydrogens in the intermembrane space. We have a very high concentration gradient of hydrogens or protons in the intermembrane space. The universe does not like things to be in high concentration. The hydrogen ions will be more stable if they diffuse, if they spread out, if they get back to some kind of equilibrium in terms of their concentration. But because hydrogen ions have a charge, they cannot get through the hydrophobic layers of the fatty acid tails of the phospholipid bilayer. The only path for these hydrogen ions to get back to the matrix to spread out into a more balanced concentration is through this last phi ATP synthase. So the hydrogen ions are going to flow through ATP synthase. As the hydrogen ions flow through ATP synthase, that kinetic energy is used to glue together a phosphate group, an inorganic phosphate group. We're going to glue that to ADP, D for di, T for tri. So this third phosphate group gets glued onto the ADP by ATP synthase using the energy from those hydrogen ions flowing through ATP synthase. This, my friends, is activated oxidative phosphorylation. We can make like 36 ATP in this process, which is amazing and keeps us alive. One more tiny little thing to throw in here. At the end of the electron transport chain, we need to make more space for more electrons. We have some oxygen. This is why we inhale the oxygen, is so that the oxygen can get to the electron transport chain. That oxygen is going to accept, it is going to take the electrons off of the electron transport chain to make more space for more jumping so that we can do this all over again. The oxygen is going to take those electrons off of the electron transport chain. Oxygen is the final electron acceptor, and that allows more stuff to happen. When oxygen gets some of those electrons and some of those hydrogens that are floating around, we end up with water. So we start with glucose. We end up with some carbon dioxide popping off of, oh gosh, where did Krebs go? Popping off during Krebs cycle. We also end up with this water here at the very end, and most importantly, all that ATP. One more time, here's our mitochondria. We have an outer membrane, we have an inner membrane. The double membrane gives us some evidence of endosymbiosis. That theory that mitochondria and chloroplasts evolved from bacteria that were engulfed through phagocytosis by really big other bacteria, and they lived a symbiotic, happily ever after relationship. So again, we've got outer membrane, we've got inner membrane, we have the intermembrane space, we've got the matrix here in the middle. What's happening in the cytoplasm out here? Glycolysis. Glucose is split into two pyruvate molecules. We also use 2 ATP to make 4 ATP, and we also reduce a little bit of NAD into NADH. The NADH and the pyruvate enter the mitochondria. They get to the matrix. The link reaction. Let me pick a different color. It's hard to see that. The link reaction is going to cause pyruvate, which is 3 carbons, to lose a carbon. That's decarboxylation. We also end up with a little bit more NADH. The 2 carbons that are left, we call this an acetyl group. It gets chunked onto enzyme CoA, forming acetyl CoA. Acetyl CoA, we're still in the matrix, is going to enter Krebs cycle. We're going to take those carbons that were left over and we're going to chop them off, and we're going to chop them off. Every time we chop off a carbon, decarboxylate this stuff, we end up with little crumbs as electrons and protons that get picked up by NAD, which is reduced to NADH. And we're also going to now add in another electron carrier. FAD is reduced to FADH2. The NADH and the FADH2 are going to carry the electrons and the protons that they pick up to the electron transport chain. The electron transport chain is embedded in the inner membrane of the mitochondria. All these folds, the cristae of the inner membrane, allow us to have extra space for more electron transport chain. The electron transport chain is going to take the electrons from NADH and FADH2, and those electrons are going to jump, 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 jump. The jumping of the electrons allows the proteins of the electron transport chain to pump hydrogens out of the matrix into the intermembrane space. So now I've got all of these hydrogens sitting in the intermembrane space. It's a high concentration gradient. This is against the laws of the universe. Those hydrogen ions want to get back into the matrix so they can spread out, get back to a more equal kind of concentration between the matrix and the intermembrane space. Their only path for getting out of the intermembrane space and back into the matrix is through ATP synthase. When hydrogen ions flow through ATP synthase, that movement, that kinetic energy of the hydrogen ions flowing allows ATP synthase to take ADP, D for 2, and one more phosphate group. We call it an inorganic phosphate group. We're going to glue those together using the energy from the hydrogen ions flowing through the ATP synthase, and we're going to form ATP, and we stay alive. Hopefully that was helpful review and not overwhelming detail.